Here is one of the strangest contradictions written into human biology. You carry something in your blood, or rather, you carry the absence of something, and that absence has haunted scientists, obsessed conspiracy theorists, and quietly baffled geneticists for nearly a century. It should not exist, not at the frequency it does, not in the people it appears in, not structured the way it is. And yet, if you are one of the roughly 7% of people on Earth who are Rh negative, your DNA is walking proof that biology does not always follow its own rules. Evolution, by most standard models, should have eliminated your blood type thousands of years ago. The logic is airtight. Rh negative blood causes a condition in pregnant women where the body attacks its own unborn child. Before modern medicine, this killed babies. Before modern medicine, it reduced the number of surviving children certain mothers could have. A trait that kills offspring should disappear. That is how natural selection works. And yet, here you are, not just surviving, but thriving in pockets of human population at rates that are scientifically anomalous. The Basque people of northern Spain and southern France carry this blood type at rates approaching 50%. Parts of the Middle East and North Africa show elevated numbers compared to the rest of the world. East Asians have it in less than 5% of their population. Indigenous peoples of the Americas and the Pacific, almost none at all. The distribution is not random. It is not uniform. It forms patterns, and those patterns point towards something very specific about where human populations came from, where they have been, and what happened in the deep history of our species. This is not a conspiracy. This is not pseudoscience. This is genetics, and what the DNA has finally started to reveal about the origin of Rh negative blood is far more complex, far more ancient, and far more fascinating than any alien theory ever managed to be. To understand where Rh negative blood comes from, you first have to understand what it actually is, and that means starting with the name, because the name is technically a mistake. In 1939 and 1940, Karl Landsteiner and Alexander Wiener were conducting experiments on blood reactivity. They injected the blood of rhesus macaque monkeys into rabbits and guinea pigs and observed that the resulting antibodies reacted with the red blood cells of approximately 85% of human subjects they tested. Those humans, they concluded, must share some version of this antigen with the rhesus monkey. They call it the Rh factor, Rh for rhesus. The 15% of humans whose blood did not react were called Rh negative because they appear to lack this shared primate antigen. Simple enough. The only problem is that it was wrong. Decades later, molecular biology revealed that the antigen found in rhesus monkeys is not the same antigen found in Rh positive humans. They are structurally related, product of an ancient gene duplication event within the primate lineage, but they are distinct proteins. The name Rh factor stuck even after scientists understood the error, because by then it was already baked into medical textbooks, blood bank systems, and the entire infrastructure of transfusion medicine worldwide. So when you hear the term Rh negative, what it actually means is not that you lack something the rhesus monkey has. What it means is that you lack a specific protein on the surface of your red blood cells that the majority of humans produce. That protein is called the D antigen, and it is encoded by a gene called RhD. The RhD gene sits on chromosome 1, right next to an almost identical gene called RhC. These two genes are so similar, sharing 98% of their nucleotide sequence, that they are thought to have originated from a single ancestral gene that duplicated itself at some point in primate evolutionary history. Both genes produce proteins embedded in the membrane of red blood cells, and those proteins are thought to play roles in the transport of ammonia or carbon dioxide across the cell membrane, or possibly in the structural integrity of the red blood cell membrane itself. The precise physiological function is still not fully understood, which is one of the reasons the evolutionary story of the Rh blood group system has remained so difficult to unravel. What causes a person to be Rh negative is remarkably straightforward at the molecular level. In most Rh negative Europeans, the RhD gene is simply gone. Both copies of it, one inherited from each parent, have been deleted. You can only be phenotypically Rh negative if you have no functional RhD gene on either chromosome. If you have even one working copy, you produce the D antigen and you are Rh positive. This means that Rh-negative status is recessive in the most literal genetic sense. You need 2 deletions, one from your mother and one from your father, to show the trait. This inheritance pattern is essential to understanding both why the blood type persists and why its distribution across the globe looks the way it does. Now we arrive at the heart of the mystery. Evolutionary biology operates on a principle that is simple in theory and brutally efficient in practice. Traits that reduce reproductive success get filtered out of the gene pool over time. Traits that help reproductive success get amplified. This is natural selection, and has been operating on the human genome for hundreds of thousands of years. By any standard calculation, Rh-negative blood should have been selected against. Here is why. When an Rh-negative woman becomes pregnant with an Rh-positive baby, which happens whenever the father carries at least one copy of the Rhd gene, the woman's immune system may eventually recognize the baby's red blood cells as foreign. Specifically, if fetal blood cells cross the placental barrier and enter the mother's circulation, her immune system can generate antibodies against the D antigen she has never encountered before. This is called sensitization. During the first pregnancy, sensitization usually does not cause immediate problems for the baby. But in subsequent pregnancies with Rh-positive fetuses, the mother's immune system is primed. Those anti-D antibodies cross the placenta and attack the fetal red blood cells. The result is a condition called hemolytic disease of the newborn, which causes severe anemia, jaundice, brain damage, and in many cases before modern treatment, death of the fetus or the newborn infant. The numbers before modern medicine were stark. Studies from the 1940s estimated that in European American populations, hemolytic disease of the newborn occurred in approximately 1 in every 56 births to Rh-negative women. Mortality among affected infants was around 20 to 40%. These are not trivial numbers from an evolutionary perspective. Every time an Rh-negative mother lost a child to this condition, the Rhd deletion allele was removed from the gene pool along with it. You would expect over thousands of generations for the frequency of the Rh-negative allele to decline steadily and continuously, perhaps not disappear entirely, because new mutations can always arise, but certainly not reach the frequencies we actually see in European populations, where the Rhd deletion allele sits at a frequency of roughly 0.43 in many studied samples. In 2012, a landmark study published in Human Genetics by geneticist Gregory Perry and his colleagues tackled this problem head-on using genomic data from 4 major human population samples. They asked a straightforward question: is there any evidence that positive natural selection has been maintaining or driving up the frequency of the Rhd deletion in European populations? In other words, does Rh-negative blood offer some hidden advantage that compensates for its cost in hemolytic disease? The answer, after exhaustive analysis using some of the most sensitive population genetics tools available, was no. There is no detectable signal of positive natural selection acting on the Rhd deletion in any of the populations studied. The allele does not carry the genomic signatures of a gene that has been favored by selection. It looks, instead, like a mutation that rose to intermediate frequency and then simply stayed there. So if natural selection did not drive the Rhd deletion to high frequency, what did? The answer that emerges from the genetic evidence is one of the most powerful forces in all of population genetics: the founder effect and genetic drift. These two forces, working together, can explain what positive selection could not. Genetic drift refers to the random fluctuation of allele frequencies in a population over time. In large populations, random fluctuations average out. A rare allele stays rare, a common allele stays common. But in small populations, the math changes completely. In a small group of humans, pure chance determines which individuals reproduce, which ones survive, and which genetic variants get passed on. A rare allele can, purely by accident, end up overrepresented in one generation, and if that generation founds a new population, the allele goes from being rare in the ancestral group to being common in the descendant group. This is a founder effect. A small founding population carries a non-representative sample of the ancestral gene pool, and that skewed sample becomes a new baseline. The Perry study found that this is the most plausible explanation for why Rh-negative blood is so common in Europeans. The RHD deletion almost certainly arose as a single mutational event, a deletion that occurred once in one individual sometime in the deep human past. Through a combination of small population sizes, population bottlenecks, and random drift, that deletion rose to intermediate frequency in the ancestral population that would eventually give rise to modern Europeans. Once it reached that intermediate frequency, around 0.43, something interesting happened mathematically. The selection pressure against it became much weaker, because when roughly half the population carries a deletion, a substantial fraction of Rh-negative women end up mating with Rh-negative men, producing Rh-negative babies who face no hemolytic risk at all. The danger only arises in Rh-negative mothers carrying Rh-positive children, and at intermediate allele frequencies, that combination becomes less dominant. The simulation models in the Perry study confirm that once the RHD deletion reaches a frequency of around 0.5 in a population, natural selection against it becomes very weak enough that genetic drift can maintain the allele indefinitely without significant pressure to reduce it. This tells us something profound about the origin of Rh-negative blood. It did not spread because it was advantageous. It spread because of who our ancestors were, small, isolated groups of people moving through a world much less populated than it is today, where every founding population was a genetic lottery, and the winners of that lottery determined what future generations would carry in their cells. No discussion of Rh-negative blood can skip the Basques, because no other population makes the mystery more vivid. The Basque people of the Western Pyrenees, straddling the border between modern Spain and France, have the highest confirmed frequency of Rh-negative blood of any population on Earth. Scientific studies have placed the frequency of the RHD deletion allele in Basques at approximately 47%, which translates to a phenotypic Rh-negative rate of roughly 30 to 35% of the population. In some earlier serological studies, estimates ran as high as 54% for the allele frequency. To put this in perspective, the global average for Rh-negative blood type is around 7% of the total human population. Among Basques, it is 4 or 5 times that. This anomaly has generated an enormous amount of speculation over the decades. The Basques are already one of the most genetically and linguistically unusual populations in Europe. Their language, Euskera, is what linguists call a language isolate. It has no proven relationship to any other known language, living or extinct, including all the Indo-European languages that surround it. Modern genetic studies confirm what the linguistic isolation suggests. The Basques have a population history that diverged significantly from the rest of Western Europe, likely representing a remnant of the pre-Neolithic hunter-gatherer population that inhabited the Franco-Cantabrian region, the area around the Western Pyrenees, during the last glacial maximum approximately 20,000 years ago. Ancient DNA studies have filled in much of this picture. The current scientific understanding is that present-day Basques are descended primarily from a mixture of pre-Neolithic hunter-gatherers who sheltered in the Franco-Cantabrian glacial refugium during the Ice Age and early Neolithic farmers who arrived in Europe from Anatolia beginning around 10,000 years ago. What distinguishes them from other Europeans is that they received far less genetic input from the third major wave of ancestry that transformed most of Europe during the Bronze Age. The Amnea steppe pastoralists who swept across Europe from upon a Caspian steppe approximately 5,000 years ago. Most modern Europeans carry significant steppe ancestry. The Basques carry very little. Their relative isolation, geographical, cultural, and linguistic, insulated them from the massive population turnovers that repeatedly reshaped the rest of the continent. A 2018 study published in European Journal of Human Genetics, analyzing both genotyping and next-generation sequencing data of the Rh gene region in Basques, Catalans, Moroccans, and several other populations, confirmed that the Basque Rh-negative frequency, while indeed the highest in the world, is not as extreme an outlier as sometimes claimed. The neighboring Catalan population shows an RHD deletion allele frequency of 41.7%, nearly as high, and populations across the Franco-Cantabrian belt, including other North Iberian groups, show a gradient of elevated Rh-negative frequencies that gradually decreases as you move away from that geographic core. This tells scientists that the Basque high frequency is not the result of some unique mutation that occurred only among Basques or some selection pressure, that affected only them. It is the end point of a geographic line, a continuous gradient that reflects a shared ancestry of the peoples who descended from those ancient Ice Age survivors. The genetic evidence points to a conclusion that is deeply human and strangely moving. The Rh-negative frequency in Basques is high because their ancestors were few, a small, isolated population weathered the Ice Age in a corner of southwestern Europe, carrying in their blood a genetic variant that would otherwise have been swamped out of existence by the larger gene pools surrounding them. They survived. They remained relatively isolated for thousands of years, and their founder effect, the random genetic fingerprint of a small founding population, persisted into the modern day. Your blood type, if you are Rh-negative and of European descent, is in some sense an echo of those Ice Age survivors. One of the most scientifically revealing aspects of Rh-negative blood is that the same phenotype, being Rh-negative, lacking the D antigen on red blood cells, arises through entirely different genetic mechanisms in different populations. This is called convergent evolution, and it is a powerful clue about the deep evolutionary history involved. In Europeans, as we have established, Rh-negative blood results almost universally from a complete deletion of the Rhd gene. Both copies of the gene are simply absent from the genome. But in populations of sub-Saharan African descent, many individuals who test as Rh-negative by blood typing do not have this deletion. Instead, they carry what geneticists call an Rhd pseudogene, a copy of the Rhd gene that is present in the genome but non-functional, rendered inoperative by a 37 base pair duplication and an inserted stop codon in exon 4. This pseudogene produces no working D antigen protein, making the individual phenotypically Rh-negative, but the gene itself is still there, broken but present. In East Asian populations, Rh-negative status is rare, around 0.5% in Chinese and Japanese populations studied. And when it does occur, it results from a variety of molecular mechanisms, including partial gene deletions and point mutations, with complete Rhd gene deletion being far less common than in Europeans. These different molecular mechanisms arising independently across human populations are significant for two reasons. First, they confirm that the Rh-negative phenotype is not the product of a single ancient mutation that spread globally from one source. Different populations became Rh-negative in different ways at different times through different genetic accidents. Second, and more intriguingly, the fact that the Rhd pseudogene is retained in African populations rather than the clean deletion seen in Europeans hints at different demographic and evolutionary histories. The African Rhd pseudogene, because it retains a gene sequence even in broken form, may reflect a different type of mutational event and a different timeline. The complete deletion in Europeans appears to have occurred as a single event, driven by a specific structural feature of the genome. The Rhd gene is flanked on both sides by nearly identical DNA sequences, called rhesus boxes. When the genome copies itself, these similar flanking sequences can misalign, causing the Rhd gene between them to be looped out and deleted, a process called non-allelic homologous recombination. The architecture of the genome in that region makes this particular deletion prone to occurring. The worldwide distribution of Rh-negative blood, when you map it carefully, reads almost like a migration map of ancient human populations, and the patterns are striking. Rh-negative blood is most concentrated in Western Europe, peaking in the Iberian Peninsula and falling off in a gradient as you move east across the continent. It is moderately elevated in parts of North Africa and the Middle East. Some studies have found Rh-negative rates of around 8 to 10% in populations from the Arabian Peninsula and somewhat higher in Moroccan samples, which may reflect both ancient migration routes and the mixed ancestry of North African populations who have absorbed input from both sub-Saharan Africa and Eurasia over millennia. When you move into sub-Saharan Africa, the phenotypic Rh-negative rate drops, typically to around 5 to 8%, though as noted, the underlying genetics are different, with the Rhd pseudogene playing a significant role. In South and Southeast Asia, Rh-negative rates drop to a few percent. In East Asia, they are under 1%. And among indigenous populations of the Americas and Pacific Islands, populations whose ancestors left Asia via the Bering Land Bridge or crossed the Pacific during a very specific and relatively recent window of prehistoric time, Rh-negative blood is virtually non-existent. That last fact is particularly telling. The near-complete absence of Rh-negative blood in Native American and Polynesian populations is exactly what you would predict if the RHD deletion were a rare variant in the ancestral populations of Northeast Asia, from which those migration waves originated. When a small group crosses a land bridge or embarks on an ocean voyage, it carries only a limited sample of the source population's genetic diversity. If the RHD deletion was already rare in Northeast Asian populations, which the current frequencies suggest, then there is every reason to expect it will be absent or nearly absent in the populations that descended from those particular founding groups. The global map of Rh-negative blood thus traces human prehistory with surprising precision, high in the descendants of ancient Western European hunter-gatherers and their admixed successors, moderate in the populations connected to Europe through ancient gene flow, and vanishingly rare in the populations most recently and narrowly bottlenecked through Ice Age Northeast Asia. No discussion of Rh-negative blood's origins is complete without confronting the Neanderthal theory, because it circulates widely and deserves a direct, evidence-based answer. The claim goes roughly like this: Rh-negative people are descended from Neanderthals, or from an admixture event between Neanderthals and early modern humans, and this ancient ancestry is what makes their blood type unusual. It is a dramatic hypothesis, and it feeds into a broader cultural fascination with the possibility that Rh-negative individuals are somehow more ancient, more alien, or more special than the rest of humanity. The actual science is more nuanced and ultimately unsupportive of this specific claim. A 2021 study published in PLOS One by Stephen Condemi and colleagues did something genuinely groundbreaking. They reconstructed the blood group status of Neanderthals and Denisovans from ancient DNA extracted from fossil specimens. Their analysis found that Neanderthals did carry RHD alleles, forms of the RHD gene, but the particular variants they carried are associated in modern humans with a heightened risk of hemolytic disease of the newborn. Intriguingly, Neanderthal RHD alleles appear to have been rare among modern human populations that interbred with Neanderthals. The study found evidence that at least some Neanderthal Rh variants were likely problematic for reproductive fitness in hybrid modern human populations, and may have been preferentially purged through natural selection in populations descended from Neanderthal modern human admixture. The headline-grabbing version of this finding, Neanderthals' blood type may help explain their demise, refers to the possibility that hemolytic disease of the newborn between Neanderthal women and hybrid modern Neanderthal fetuses may have been a contributing factor in Neanderthal extinction. What this does not mean is that Rh-negative modern humans carry Neanderthal blood types, or that being Rh-negative is a sign of Neanderthal ancestry. The RHD deletion that causes most modern Rh-negative phenotypes in Europeans is not the same thing as the Neanderthal RHD variants identified in ancient DNA. They are different variants within the same gene system, arising from different mutational events with different functional consequences. The Neanderthal connection to the Rh system is scientifically real, but it is not the connection the popular narrative usually describes. The science here is genuinely interesting and complex, but complexity does not survive well in online speculation, where it tends to get flattened into a simpler, more dramatic story. At this point, it would be dishonest not to directly address the landscape of claims that have grown up around Rh-negative blood, because many of the people watching a video like this have encountered them and are trying to figure out what is true. The claims range from the merely unsubstantiated to the flatly contradicted by evidence. The most persistent claim is that Rh-negative blood is associated with specific physical traits: green or blue eyes, reddish hair, higher IQ, lower body temperature, heightened psychic sensitivity, empathic abilities, a sixth sense for danger. None of these associations have been established in scientific literature. Blood type is determined by specific proteins on red blood cells. It does not cascade to the body, influencing eye color, hair pigmentation, or neural architecture in any documented mechanism. These associations are built on confirmation bias, the very human tendency to notice evidence that fits a pattern and ignore evidence that doesn't, amplified by internet communities where people who share both a blood type and a belief system reinforce each other's observations. The extraterrestrial theory, the claim that Rh-negative people are descended from alien beings or interdimensional entities who interbred with humans, is not falsified in the technical scientific sense, because it is not a scientific claim. It does not make testable predictions. It does not offer a mechanism. It is, in the strict sense, unfalsifiable mythology. But it is worth noting that everything we do know about Rh-negative blood, the genetics, the population distribution, the molecular mechanisms, the evolutionary history, is fully and elegantly explained without reference to anything supernatural or extraterrestrial. The actual story of where the RHD deletion came from, how it survived, and why it concentrates in the populations it does is sufficiently extraordinary without embellishment. There is also a claim, echoing in various forms, that Rh-negative blood dates to approximately 35,000 years ago, when it supposedly appeared fully formed in a population with no prior history. This is not an established finding. Population geneticists have not pinpointed a single origin date for the RHD deletion with that kind of precision. Mutation dating is a probabilistic exercise that depends on assumptions about mutation rates in population history, and current evidence suggests the deletion may have arisen at various times through the structural propensity of the rhesus box flanking sequences, potentially more than once, in different populations at different times. One intriguing hypothesis that does have some scientific backing involves a tiny single-celled parasite called Toxoplasma gondii. This is the organism responsible for toxoplasmosis, a globally widespread infection that infects roughly 1/3 of the world's human population, usually with mild or no symptoms, but with significant effects in immunocompromised individuals and developing fetuses. A series of papers by Czech researcher Jaroslav Flegr and colleagues have suggested that Rh-negative individuals may respond differently to Toxoplasma gondii infection than Rh-positive individuals, and specifically that the Rh factor status may modulate some behavioral and physiological effects that Toxoplasma has on its human hosts. Flegr's research, which remains controversial and has not been conclusively replicated across all populations, suggests that Rh-negative individuals show different patterns of toxoplasma-associated changes in reaction time, certain behavioral tendencies, and even some health outcomes. The proposed mechanism is not fully understood, but the hypothesis is that the Rh proteins embedded in the red blood cell membrane and potentially involved in biochemical transport functions may interact in some way with the immune response to toxoplasma infection. If Rh-negative status conferred some advantage in resisting or tolerating the parasite, even a subtle one, this could represent a counterbalancing selective force that helped maintain the RHD deletion at higher frequencies than pure selection against hemolytic disease would otherwise allow. This is speculative, and the Bass study published in European Journal of Human Genetics in 2018 noted explicitly that this association has not been clearly demonstrated. But it represents exactly the kind of legitimate scientific hypothesis that the field needs, one that treats Rh-negative status not as an anomaly to be explained away, but as a functional biological variant that may have consequences we do not yet fully understand. The Rh proteins do something in the red blood cell membrane. The function is not well characterized, and until it is, the evolutionary puzzle of why the RHD deletion persists at the frequencies it does in European populations will remain at least partially open. So let's bring this back to you, personally, if you're one of the roughly 500 to 600 million people on Earth who carry Rh-negative blood. What does the science actually say about where you come from? If you are primarily European descent and Rh-negative, your blood type is almost certainly the product of an RHD gene deletion that arose sometime in the deep history of the populations ancestral to modern Europeans, rose to significant frequency through genetic drift and founder effects in small prehistoric populations, and was carried forward into the present day by the demographic accidents of human prehistory. You are not a descendant of aliens. You are not the product of Neanderthal interbreeding in any way that is specifically unique to Rh-negative individuals. You are, in the most literal genomic sense, a descendant of Ice Age survivors, the people who sheltered in the refugia of southwestern Europe during the last glacial maximum, who emerged when the ice retreated, who absorbed some of the genetic input from later migrating populations, but retained, in their blood, the molecular signature of their ancient isolation. If you are of Middle Eastern or North African descent and Rh-negative, the picture is similarly rooted in ancient population movements and mixing events that played out across the Levant and the Saharan corridor over tens of thousands of years. If you are of African descent and Rh-negative, you may carry the RHD pseudogene rather than the complete deletion, and your ancestry traces a different molecular path to the same phenotypic outcome, a path shaped by the extraordinary genetic diversity that accumulated in Africa over the longest span of human evolutionary history. And if you are of East Asian or Indigenous American descent and Rh negative, which would be genuinely rare, you likely represent a recent mutation or an exceptional genetic inheritance from populations that mix with European or Middle Eastern source populations within the historical or late prehistoric period. None of this is as dramatic as an alien bloodline, but it is something arguably more profound. It is evidence of the long journey that every human population has taken across this planet, carrying its genetic history in every cell, including the red cells that move through your veins right now. Your blood type is not a cosmic mystery. It is a human story, ancient, complicated, and written entirely in the language of DNA. It would be intellectually dishonest to close without acknowledging what remains genuinely unknown. Honest science does not pretend certainty where uncertainty persists, and the Rh blood group system has several genuinely open questions that researchers are actively working to answer. The precise function of the Rh proteins in red blood cell physiology remains incompletely understood. They are clearly important. Humans with the extremely rare condition known as Rh null blood type, in which all Rh antigens are absent, have fragile red blood cells and suffer from a mild hemolytic anemia, but the exact molecular roles of the D antigen and the Rhc proteins in membrane structure and biochemical transport are not yet fully characterized. Until they are, it is difficult to construct a complete and definitive model of why particular variants of the Rh system were selected for or against in different populations. The question of whether any form of balancing selection, where both the presence and absence of the D antigen have advantages in different contexts, has ever operated on the Rhd locus is still technically open. The Perry study found no evidence of positive selection, but the statistical tests used have limited power to detect old or complex selection events, and a recent preprint from 2026 analyzing quantitative models of Rh negative allele frequency suggested that statistically anomalous frequency peaks in geographically linked populations may merit further investigation. The field is still working on this question with improved tools and larger datasets. The relationship between Rh status and infectious disease resistance or susceptibility, including the toxoplasma hypothesis and other proposed immunological interactions, deserves more rigorous investigation than it has received. These are not fringe ideas. They are legitimate scientific hypotheses that have appeared in peer-reviewed literature and that deserve the kind of large-scale, well-controlled studies that might finally resolve them. What the DNA has revealed so far is substantial, and it is extraordinary in its own right. What it still holds may be more extraordinary still. Your blood type carries a record, not of alien civilizations or fallen angels or ancient curses, but of human beings who walked through ice ages, crossed mountains, founded small communities in isolated valleys, and passed something forward through generations without knowing what it was or why it mattered. The RHD deletion in your genome is one of the quieter chapters in the story of our species. Quiet because it survived not by being loud or powerful or advantageous in any obvious way, but simply by being there in a small enough group at the right time, carried forward by chance and by life and by the stubborn persistence of human populations through every climate catastrophe, migration event, and population bottleneck the last 100,000 years could throw at them. That is where you come from. That is what your blood is telling you. And that story, it turns out, needs nothing added to it.